Indeed, let's move on to the last talk in this afternoon's session, and that's from Francesca Coscia, who did her PhD in an old lab mate of mine with Guy Schoen and colleagues in Grenoble. She's since been a postdoc at Birkbeck and at LMB here in the UK, and is now a PI at the Human Technopole in Milan, and is going to tell us about the structure of human thyroglobulin. Francesca. We can see your
Thank you very much. Can you hear me? Can you hear me? Okay, thank you. Thank you, Neil, for the nice introduction, and thank you, the organizers, for inviting me. So today I'm glad to tell you about the story of thyroglobulin structure determination, which was performed by single particle cryo-EM during my postdoctoral work at the MRC LMB in the group of Jan Löwe. So first of all, what is thyroglobulin? It is the main protein of the thyroid gland, which is an organ located in the neck of all vertebrates. Now, the structure of the tissue of the thyroid is composed by follicles, which are monolayers of thyroid cells surrounded by blood vessels and enclosing a large extracellular space. So here, thyroglobulin is secreted at extremely high concentrations, so you can extract a lot of thyroglobulin from a thyroid. And because of this, and because this is a very stable, large dimer, it has been used since the early ages of biochemistry as a molecular weight standard. So biologically, thyroglobulin is the precursor of thyroid hormones, which are two iodinated molecules essential for the metabolism of vertebrates. So thyroid follicles are basically some small bioreactors which accumulate the rare iodine from the diet. So all the iodine that we eat is concentrated in the thyroid, as you can see in this iodine reactive iodine scan. And then the thyroid uses thyroglobulin to make those two hormones. So we have two types of thyroid hormones that are secreted in the bloodstream, T4 and T3. T4 with 4 iodine atoms and T3 with 3 iodine atoms. So despite the majority of the hormone produced is T4, T3 is the most active form on metabolism. And in particular, from the bloodstream, those two hormones reach basically every cell of the organism, and they activate the transcription of metabolic genes controlling fundamental processes such as development and growth. Now, the important parameter is the T4 to T3 ratio, which is modulated across different species depending on the metabolic needs, but also across the lifespan of the same organism. So the hormone imbalance implies severe human diseases, and unsurprisingly, TG mutations are linked to hormone impairment. Now, despite extensive studies in the field, controlling T4 to T3 ratio in vivo is still a challenge. And so we wanted to know more about how thyroglobulin actually synthesizes those two hormones. So as you can see here, they are made of a tyrosine backbone. And the reason is because they are made by an unusual mechanism from the own tyrosines of thyroglobulin. So if we take a closer look at the follicle, here we have the thyroid cells surrounded by blood vessels and the extracellular lumen. So here the iodine is in its reduced form is uptaken, and then TG, thyroglobulin, is secreted. So its tyrosines are enzymatically iodinated, and then an interside chain rearrangement reaction happens by which one ring is transferred to the other, thereby synthesizing the hormones directly onto the polypeptide chain of TG, in this case T4, in a position which is called ring acceptor, and leaving an empty side chain or a dihydroalanine at the donor position. So at this stage, the hormones are still attached to thyroglobulin, and to be released, thyroglobulin needs to be internalized in the cell and then proteolyzed in the lysosome to allow the release into the bloodstream. So out of about 120 total tyrosines in TG, about 60 are iodinated, and only a very small number synthesizes hormones. So the positions of the acceptors was partially identified when we started this work, while the position of the donors was still uncertain. But most of the other iodotyrosines just serve as an iodine reservoir, so they do not form hormones. So the mystery here was to understand why the first tyrosines are so special. And here our interest of determining thyroglobulin structure and then understand how its fold brings together those two special tyrosines forming hormones. So over the years, it has been quite challenging to determine thyroglobulin structure, although it's quite abundant, it's a very well-known protein for a number of reasons. First, because it's mainly extracted from thyroid, so it can be quite heterogeneous, and also because it's highly glycosylated, so it is difficult to get well-diffracting crystals. Moreover, it is composed of many repeated sequences, so it is actually very hard to find macro domains to do crystallization of parts of it. So our goal here was to use single particle cryo-EM to solve the structure of thyroglobulin, identify those special hormogenic sites, and understand more in depth the mechanism of hormone synthesis. So we started with the endogenous protein from patients. This is obviously partially iodinated, contains variable amounts of hormones, and batches are quite variable. It is also partially degraded, but that at the same time, in collaboration with Veronica Chang in the group of Radu Aricescu, we produced TG from HEC cells. And this is much more homogeneous and reproducible in preparation, is completely uniodinated, and of course it's much easier to do mutations. So the first EM imaging of thyroglobulin happened in the 80s, and here you can see this beautiful negative staining EM micrograph. So with the human thyroglobulin, this is from rat thyroglobulin, we tried to reproduce this, and it worked straight away for negative staining. But unfortunately, it was really challenging to get good cryo-EM grids for high resolution data collection. So as you can see, for unsupported eyes, the protein was completely denatured at the air-water interface. So after testing a number of conditions, our golden combination was to use a mild deglycosylation and graphene oxide supported grids. So this procedure worked for both endogenous TG and recombinant TG. And as you can see from the 2D class averages, they really look similar. This was a very important step in the project because it means we can perform biochemical studies on recombinant TG with confidence. So then we collected two data sets for endogenous and recombinant TG, one the highest resolution, one at EBIC, and then one at the LMB, using quantifoil grids covered with the graphene oxide. These were collected in very similar conditions, and then we imposed C2 symmetry to get a first map in Reliant. So the overall resolution is 3.5 angstrom, but as you can appreciate from the map, so this is quite much lower resolution in the peripheral regions. And because we didn't have any crystal structures, we really needed to improve the map in the peripheral region to do de novo building. So in order to do this, we used, it has been mentioned today already, and I'm sure that many of you are familiar with symmetry expansion. We used a combination of symmetry expansion and recentering to use to improve the local map quality. So if we have the n initial particles where we have the information doubled into two lobes A and B, we just take the star file with the n particles and we apply C2 symmetry. Then we combine those two data sets. So in the two parts, we will have the information replicated A, B, and BA. So we only need one lobe. So we will apply a mask, and then this is already advantageous because we can align B to A and improve the resolution, perform 3D classification. And then further, to improve the quality of the map, we also recentered into the most flexible part. And this led to a massive improvement of the map that then allowed the de novo building. This was really challenging because, as I mentioned earlier, TG is made by many repeated sequences, and they are also cysteine rich. So it is actually quite difficult to find the right path of the polypeptide chain. And the only big domain is a dimeric cholinesterase-like domain at the C terminus. So using a combination of tools, many of them I've learned at the ICNIL CCP-EM workshop. So we managed to build 93% of the sequence, and this giant dimer has about 120 disulfide bonds and more than 30 glycosylation sites. So here is an overview of the thyroglobulin dimeric architecture. This is actually an intertwined dimer with an extremely large dimeric interface. And despite we deglycosylated the protein, we could resolve many unlinked glycans. And interestingly, some of them are at the interface between two subunits. So they are actually part of the structure and stabilizing the dimer. So now if we color the sequence in a rainbow color scheme from the N blue to C terminus red, then we can place, we could identify some large macro domains. So if we start with the N-terminal domain, NTD, we see that then the polypeptide chain crosses the C2 axis. We have a core, a flap, an arm, and then a dimeric C-terminal domain that folds back. So as you can see here, the structure is very entangled, and the C-terminal domain basically interacts with all other macro domains, which are very, very far in sequence. And by the way, both PDB and the data sets are available, so feel free to play with it and experiment. So because this is a very intricate structure, we wanted to verify it with a parallel technique. And so we used cross-linked mass spectrometry in collaboration with the group of Yuri Rapsilba to verify the structure. So first we cross-linked TG with the BS3, and this resulted in a beautiful dimer, a stable dimer, so there were no aggregates. And then from the structure, we predicted the theoretical cross-links, and then we overlapped the experimental cross-links. So now to verify this intricate structure, we are not interested in the ones along the diagonal, which are interactions between residues very close in the sequence. But the most interesting are far from the diagonal, and these were very important to verify the structure. Not only we could verify the proximity of residues that we could actually see and resolve in the map, but also some that were not resolved. So after we solved the thyroglobulin structure, we came back to our original question. So what are the hormonogenic sites, and what is the mechanism of hormone synthesis? So as I mentioned earlier, the acceptor position where the hormones are found were identified by mass spectrometry quite recently, and while the position of the donors was quite uncertain. So the hormonogenic sites are historically called site A, B, C, and D, and we managed to resolve 3 out of 4. So what we did is to find the acceptors into the structure and then look in their proximity to find the possible donors. So site C was not resolved because it's a very C terminus. And then in site A, for example, we have 1 acceptor and actually 2 possible donors. This happens within the same subunit. while for site B, we only have one possible donor that could pair forming hormones. And this is always within the same subunit. And then in site D, we have a single donor, but this time across two different subunits. So we identify this from the structure, but actually we wanted to verify experimentally whether the sites we found are actually forming hormones. So from this analysis, we have 4 acceptors and 5 donor types. So we developed an in vitro iodination assay. I wouldn't go into detail, but basically we try to reproduce what happens in vivo. So we iodinated completely uniodinated recombinant TG, and then we liberated the hormone from TG, and then we detected it with the ELISA method. So in order to verify the identity of these tyrosines, we used the recombinant TG and we mutated the tyrosines into phenylalanine in order to suppress the hormone formation. So we did this initially with T4, which is the major product. And here is the T4 formed by the wild type TG. So if we mutate all the acceptors into phenylalanines, then the activity is completely suppressed. And similarly for the 5 donors. So there are no more than 4 acceptors and 5 donors. However, in site A, and this was not predicted, we have 2 different donors. So in order to verify this, we produced 2 different mutants in which one of the donors is present at a time, and then we have some residual activity. So it means there are actually 4 acceptors and 5 donors type. And with this experiment, we unambiguously determined the stoichiometry of hormone formation from thyroglobulin. So to summarize, we have 2 copies of site A, B, and D, and 1 copy of site C, which is across 2 subunits from the same tyrosine. So at this point, we wanted to, since these sites are in very different domains, we wanted to compare the chemical environment and to look for some determinants or geometrical constraints for the hormone formation. Yet, to our surprise, after extensive mutagenesis and structural analysis, the only common features that we found is that the 2 tyrosines are proximal, they are exposed, and they're found in extremely flexible regions for efficient coupling, probably. So, but now the question is what all the other tyrosines are doing. So why aren't they forming hormones? And so from the structure, we calculated all the possible tyrosine pairs and verified whether those 3 determinants were satisfied. Well, we found that some of the close tyrosines are too buried to be iodinated and some are too far apart to react. So basically all the other tyrosines are isolated and cannot pair, but they just serve as an iodine storage device. So again, we confirmed there are only 4 tyrosine pair types within thyroglobulin that can actually generate hormones. So the conclusion is that there's actually no structure active site we were looking for. But this also means that in principle, we could make thyroid hormones from any other protein that has 2 flexible tyrosines. And so in order to test this hypothesis, we considered another famous well-known protein in the lab, the maltose binding protein, which is surprisingly rich in tyrosines. Most of them are buried, but some of them are exposed and they can be engineered to test the hormone synthesis. So in particular, we have in the wild type a beta hairpin here containing 2 tyrosines, which could form hormones. And then we have a very exposed Y341, which is isolated, but we could create a pair by mutating this arginine or adding a peptide at the C terminus, which is quite close. So we produced the different variants of MBP, and then we tested the T4 production following the same method that we had for TG. So for the wild type MBP, we couldn't see any hormone formation. And this is probably because the 2 tyrosines are proximal, but not quite exposed or flexible. And same for these two tyrosines, because they are constrained in rigid alpha helices, so they are proximal, exposed, but not quite flexible. So then we played with flexibility, adding C-terminal peptide to pair with the Y341. And in this case, we certainly have exposed tyrosines are quite flexible, but not quite close. And that's why we still didn't have hormone formation. But then if we elongate this peptide, we strikingly managed to obtain hormone production with the same efficiency of 1 TG site. And this is because all the three requirements are met. So the tyrosines are proximal, exposed, and flexible. And then to push this forward, we asked whether even tyrosine copolymers could form hormones. And this is the case if we work at the extremely high concentrations. So in summary, in this work, we determined the first cryo-EM structure of human thyroglobulin, and this is very well conserved in all vertebrates. So this covers a huge importance biomedically, because now we can map disease mutations in 3D, and we can also measure the synthesis defects in vitro using our assay. So thyroglobulin from our studies is a giant scaffold substrate, and its tyrosines are iodinated. So when they are isolated, they just serve as an iodine storage device, while if they are close to each other, then they form hormones. So probably the most important outcome is that we determined unambiguously the stoichiometry of so how many copies of hormones are formed from TG. But while we know how many copies of the hormones are formed, we don't know anything about the ratio between T4 and T3. And in fact, from in vitro iodination of TG from HEK cells, we couldn't detect any T3, which is the metabolically active hormone. And with our MBP experiment, we proved that basically any protein could form T4. So now the question is, why do we need TG at all, and why this giant multi-domain protein has been conserved over evolution if a much simpler protein could exert the same function? So this probably lies beyond the iodination reaction itself, and it's due to the many regulatory processes in which TG is involved, and that couldn't be exerted by a much simpler protein. In particular, in the extracellular lumen, thyroglobulin is able to regulate, to control a T3 to T4 ratio, and then to control the hormone release by receptor-mediated endocytosis, and also to provide an efficient storage of the iodine, is able to cross-link in a dense extracellular matrix. So we are, with this structure, just at the very beginning of understanding all this process. And in my group at the Human Technopole, I'm going to investigate in depth these aspects. So there's much more to be discovered on TG. And we believe that cryo-EM, given the extracellular and heterogeneous nature of the proteins involved, is the best way to address these long-standing questions in thyrobiology. So we will use single particle cryo-EM, but also in situ cryo-electron tomography. So there are some PhD and postdoc positions available, and I invite you to apply and to contact me if you're interested in the topic. The goal of the overall research is not only to understand the hormone regulation and obviously the implications in human diseases, but also to start to study the evolutionary origins of iodine and thyroid hormone biology, which is a quite uncharted area in biology. So with this, I'd like to conclude, and I would like to thank all the people that work with me at this project, and wonderful environment of MRC LNB, and my supervisor Ian Löwe and his group, Radu Aricescu and Veronica Chang, and all this group that helped me with the HEK cells production of TG, LNB, scientific computing, all the people that helped me with the model building, and also the ICNIL CCP-EM workshop, which was quite useful. Of course, the people that helped me collecting data at the LNB and ABIC, Dan Claire, source sharers for support with the image processing, and my collaborators at University of Ljubljana, Berlin, and Bristol for their help. And then finally, I'd like to thank Human Technopole for hosting my lab and to allow me to continue this exciting research. And thank you all for your attention.
Thanks, Francesca. What a lovely talk. So I've got a quick question. So I'm sorry if I missed this, but so is there how does TG get iodinated? Is it simply a solution chemical reaction, or is there a cofactor? Or is it just driven by enrichment of iodine into the tissue?
Yes, so I'm gonna, if you allow me, I'm gonna come back to that slide. I went quite fast on it.
Yeah, sorry.
So, so there's actually here, there's a iodine, sodium iodine is imported that specifically pumps iodine inside. So from the bloodstream into the thyroid follicles, and then in the, this reaches the extracellular compartment together with TG, and then this is still in the reduced form. So then there are two enzymes, duoxidase that produces hydrogen peroxide, and then iodine is oxidized, and then TPO peroxidase catalyzes the iodination of iodine of tyrosines in thyroglobulin. However, the same reaction can also happen just with the iodine. So exactly as you do with the crystallography when you derivatize.
Yeah, so I mean, the reason I asked is I was just a bit surprised that the non-active tyrosines act as a reservoir, because how do you get the tyrosine off them?
Sorry, how do you get?
Yeah, how do you get the tyrosine off your storage tyrosine? How do you get the iodine off your storage tyrosine?
Yes, that's an excellent question. Yeah, sorry, I didn't include all the cycle. So actually, in the cell, there are some deiodinases that remove the iodine and then recycle this back to the, so this is recycled, but then partially this is also accumulated extracellularly in this highly cross-linked thyroglobulin. So when the protein is undergoes endocytosis, the tyrosines that form hormones, they are just liberating hormones, and then these are secreted through a system of pumps in the bloodstream, and then the iodotyrosines, which didn't form hormones, they are deiodinated, and then they are recycled. The iodide is recycled back.
Okay. All right, great, thank you. So there are some questions in the chat. So first one from James is, was there much conformational heterogeneity in your data that you classified away?
Yeah, there was some, and it was mostly in endogenous, obviously, thyroglobulin. So here's a 3D classification. And so basically, so this is after the symmetry expansion. So this corresponds to the N-terminal domain. And so there were a few classes in which it was quite flexible. So I only selected the best ones and then refined them. We still don't know what the other ones do. So I tried to produce a high resolution reconstruction, but this was just, with a flexible N-terminal domain. And this might have some functional implications, but we still don't know this.
Yeah, then there's a follow-up question which is related to what you just said. You might want to just add a little bit. Is there any reason applying the C2 was not enough to get high resolution and you had to go for symmetry expansion?
Yes, so if I just apply, since it's elongated, so the extremities are flexible. So yeah, I first applied it. So the question is if I could do just C1, is that the question?
The question is there any reason applying the C2 was not enough to get high resolution and you had to go for symmetry expansion?
Well,
not sure.
Well, I tried, yeah, I mean, it's a dimer, so you want to apply C2. If you, so just a second, I'm trying to find the slide back. Okay, yeah, so if you apply, if you don't apply C2, then the map it's at lower resolution, is about 4. So that's with the C1.
So it is genuinely symmetric.
Yeah. It's yeah, already from the 2D class averages, you can see this. It's really a dimer. And then, yeah, so you need symmetry expansion, because you could do just masking, but then you will lose half of the information. So you want to use the information for both lobes. So that's why I did symmetry expansion. So you have the information of the lobe of the subunits B and A on the same side. So then you can mask this, that you can align them to each other and then perform 3D classification. And then this was improving the map already, but then the big improvement was recentering in the peripheral region, because then the angles are determined based, with respect to the new center.
And then I'm going to jump back in with a question of my own before asking the last question in the chat, which is that you showed that some negative stain of your crosslink material, which looked beautifully monodispersed. I wondered whether you actually solved the structure of that as well and compared it to your...
Yeah, so I didn't, but actually, so it was working very well in negative staining, but not so well in cryo. Okay. So probably because, yeah, the surface was modified. Yeah. So this would have been, I actually tried, yeah.
And then the last question is, again, a biological one. Very nice talk, thank you. Is from Alessandro Borsellini. Is there a reason why you didn't use the iodinated TG to see where the hormones, to actually see where the hormones are formed rather than the...
Yes, so this is in the plans. This is in the plans because, I mean, initially we wanted to have much more homogeneous thyroglobulin. So if you extract this from natural sources and from patients, it's always partially iodinated. You never control exactly how much is iodinated. So one should do controlled in vitro iodination, try to reproduce exactly what happens in vitro. And then you should classify out the iodinated from, uniodinated. So this is in progress. It's on the to-do list. But yes, it will be very interesting to see the hormones bound to it.
Okay. I think we are done. So as chair, it just falls to me to thank all of the speakers for a set of fantastic talks. Thanks, Francesca. And if there is nothing from CCP-EM guys, we'll draw the session to a close, and I look forward to seeing you all tomorrow.